Hi everyone, we will have lab exam 2 exercise today. And this lab exam 2 will be over lab experiments 11, 12, 13, 14, 15, 16, 17, 18, 19, and 20. And most of these experiments are used for identifying the unknown bacteria. Okay, so instructions for the best results of this exercise. Make sure you repeat lab experiment 11 until 20 as many as possible before taking this exercise. It will help you a lot to understand the exercise. Prepare some pieces of paper and pen or pencil to write your answer. There will be 2 or 3 seconds after each question to write your answer on paper before the answer key is given and explained. Remember, you can always pause the video if you need more time. After finish the exercise, check your answers, how many are correct and how many are incorrect. Repeat the process until you get 100% correct answers. Make sure that the 100% correct answers are based on your knowledge, not from guessing. It is very likely that you will get a very good score on your lab exam number 2 if you follow this instruction correctly. Okay, for this exercise or this lab experiment 11 until 20, you have to sure that you able to use the dichotomous key, which is the key used for testing unknown bacteria. So the first thing that you do is actually to isolate the bacteria until the pure culture. And then this pure culture is the one that will be identified with this test. The first test that done will be the gram stain, okay, to know whether the bacteria is gram positive or gram negative. Okay, and then after that, if it is gram negative, there will be more tests, like for example, oxidase test. Okay, and then after that, there will be another test. So look at the second dichotomous key over here. So let's say we already test that the bacteria is actually gram-negative bacteria. Then the next test will be oxidase test. And if the test is negative, then the next test will be lactose fermentation test. So you have to follow this test in order to identify the unknown bacteria. Let's say in the lactose fermentation test, you get positive result. Then what you have to do the methyl red test. That's the next test after fermentation, lactose fermentation test. In the methyl red, let's say you get positive result, then what you have to do the SIM test. One of them is an indole test. And if it is positive, then you will sure that the bacteria is Escherichia coli or E. coli. If it is negative, then the bacteria will be Citrobacter frundii. So this is how to use this dichotomous key. And this is the one that you use for all the experiment 11 until 20. And it is very important key to identify this bacteria over here, the unknown bacteria that you have. Which one is that? Whether Vibrio cholerae, Pseudomonas aeruginosa, or maybe Shigella. So this is very important. Make sure you're able to follow this key in order to know the unknown bacteria. Okay, we start. Okay, we start with question number 1. Cell shape arrangement, presence of flagella, colony arrangement, and presence of endospore are examples of blank characteristic that can be helpful in identification of bacteria. So what kind of characteristic is this? Cell shape arrangement. So yes, choose your answer. Yes, it should be the morphological characteristic. Its shape, arrangement are morphological characteristic. Okay, so that should be the answer. Number 2, using antibodies to detect the presence of antigens is an example of blank characteristic that can be helpful in identification of bacteria. Okay, what is this? A, B, C, or D? Okay, remember antibodies, antigen reaction. It is a type of immunological test. So this should be the answer for this question. Number 3, what is the most widely respected resources for bacterial identification? So make sure you read your book, make sure you follow the lab experiment. It is mentioned in one of them or some of the experiment. This is the Burgess Manual of Determinative Bacteriology. This is the most valuable book for bacteria identification. The next one, the Burgess Manual Determinative Bacteriology are based on what? So what it is based on? Genetic, immunological, morphological, or DNA? Yes, it should be based on the morphology and physiological characteristic of bacteria. That's the identification in Burgess Manual Determinative Bacteriology. Which of the following must be done before attempting to identify a bacterial species? Which one is that? I already mentioned to you in the introduction of this exercise. Yes, you have to isolate first. Make sure it is pure culture that you will identify. Do not, use mixed culture because it's going to be difficult. Okay, make sure it is pure culture. So you have to isolate that bacteria first into a pure culture. And you know how to do it on the previous lab experiments, okay, lab experiment group 1, okay, before the midterm. Okay, after the unknown bacterium has been isolated in the pure culture, what is the first step in the identification process? Remember the dichotomous key. What is the first test what we have to do? A, B, C, or D? Yes, the first test that we have to do is gram staining in order to know whether the bacteria is gram positive or gram negative. Next one, gram stain alone can identify many bacteria. Yes or no? It is false. Okay, gram negative, gram positive, there are so many bacteria in this group. So gram stain alone cannot really identify the bacteria itself. It just identifies the group of bacteria, whether positive or negative. Gram-positive bacteria show purple color under the microscope after stain with the gram stain. True or false? Yes, it is true. Okay, so after gram staining, use the microscope. If you see blue or purple color bacteria, that bacteria is gram positive. If it is red or pink color, it is gram negative bacteria. Okay, so this is one. So this one is gram positive bacteria. So this is gram positive bacteria. And this is the gram negative bacteria, the color that you see under the microscope. microscope. Okay, gram-negative show purple color under the microscope after stain with the gram stain. Of course, the answer is false. Remember, gram-negative bacteria will be red or pink color. Gram-positive will be blue or purple color. The primary stain used in gram stain is the A, B, C, or D. What is that? The primary stain used is crystal violet, which has a purple color. Okay, so this is the primary stain. The secondary or counter stain is the safranin. It has red color. Okay, so if you see this, this is the first thing that we have to do when gram staining is using the primary stain, the crystal violet, which is blue or purple color. And then the last step is using the counter stain, which is safranin. It has red color. So if we perform this test correctly, then, okay, if your bacteria is gram positive, then it will be blue. So if you see the round or coccus bacteria over here are gram positive. The coccus is gram positive in this test. And the bacillus, okay, bacteria, the rot bacteria, is gram negative because the color is red. Okay. The counter stain used in gram stain is the A, B, C, or D. I just mentioned that. The answer will be safranin. And safranin carry red color. Gram negative will retain the safranin. Gram positive bacteria will retain the crystal violet color. The gram positive bacteria have blank cell wall or peptidoglycan that retain the blank color. Okay, what is the answer for this one? Yes, gram-positive bacteria have thicker cell wall. And this thicker cell wall or the peptidoglycan will retain the primary stain. Okay, primary stain, which is crystal violet, which is purple color. Okay, so it's thicker and retain the primary stain. Okay, so this is gram positive. You see it's purple. Okay, and then gram negative, it is red color. And you see this one, the gram positive bacteria has thicker cell wall, while the gram negative, it has thinner, okay, cell wall. Now the next test is the after gram positive or gram negative or gram staining test. The next test is oxidase test. The oxidase test detects the presence of enzymes blank, which is an important component of electron transport chains in aerobic cellular respiration. So it is used for aerobic cellular respiration. This enzyme is used for this aerobic cellular respiration. It's especially on the last step, which is the electron transport chains. So you have to know the cellular respiration that starts with glycolysis, Krebs reaction, Krebs cycle, and then the electron transport chain. So what is the name of the enzyme? Yes, it's mentioned over here. Okay? The name of the enzyme is called the cytochrome oxidase. So this test will detect the presence of cytochrome oxidase If this enzyme present, then the result will be positive. It is not present, then the result will be negative. The oxidase test requires the use of reagents called blank reagents. What is the name of the reagents used for this oxidase test? A, B, C, or D? Yes, so we're gonna use the tetramethyl P phenyl diamines. Okay, it's actually gonna drop this reagent, okay, on the swab. Okay, the swab that has the unknown bacteria. Okay, so the reagent called the tetramethyl P phenyl diamines. Now, in the oxidase test, if the column is selected with a swab and the addition of oxidase reagents result in no color change, is an indication of blank bacteria, oxidase positive or oxidase negative. Of course, if it is no color change like this, it's going to be oxidase negative. This means that the bacteria do not have the cytochrome oxidase enzyme. In the oxidase test, if the colony is selected with swab and the addition of oxidase reagents result in dark blue color, it is an indication of blank bacteria. It is indication that bacteria have or able to perform that reaction. It has cytochrome's enzyme, and it's going to be called the oxidase positive over here. So blue, dark color, positive. Which of the following bacteria is unknown bacteria of lab experiment 11, unknown sample 1? So if you perform experiment 11, you should be able to answer this question. Which show oxidase positive results. Okay, so after gram is gram negative, and then the oxidase is positive. Non-glucose fermenter is mean that negative, unable to ferment the glucose, and the nitrate reducer is positive result. So which one is this? Okay, the answer is this, but you will have this dichotomous key to answer that question. Okay, which is, okay, remember the first test is gram negative, gram staining, and the result is gram negative. What is the next test? The next test will be the oxidase test. And the oxidase test is, say, positive result. So positive means this one. Okay, it means that it's glucose fermenter. Next, okay, the test is glucose fermenter or not. Okay, so this is it's non-glucose fermenter. It means that it's negative. Okay, so from here going here, so negative. And after that, you have to perform the nitrate reduction test. And then what is the result? It says that the nitrate is reducer, which is positive. So this one over here. Okay. Now, because positive, then you're going down, you get the answer, which is the unknown bacteria is Pseudomonas aeruginosa. Okay, that's how to find the unknown bacteria based on the tests, several tests that we perform. Okay, the cytochrome oxidase enzyme is the key enzyme catalyzing reactions in the lactic fermentation pathway. True or false? Okay, remember that cytochrome oxidase is used for aerobic cellular respiration. It is not fermentation. So it's gonna be false. Okay, cytochrome oxidase is used for aerobics, cellular respiration. Remember, fermentation, it is anaerobic. Okay, so that's make it wrong. It is not the correct answer. The phenol red glucose broth with Durham tube is used to determine bacteria that can A, B, C, or D. Choose your answer. It's used for determine bacteria that are able to ferment glucose. Okay, that's the phenol red glucose. It's actually mentioned over here. Okay, it's a glucose fermentation. So this is the result over here. So the result of the test, if it is negative, then the color stay the same. So this is negative. Okay? If it's positive, then the color will be yellow because it's become acidic. Okay, acidic. This one is neutral. Okay, so when the bacteria are able to ferment the glucose, then the color will change from red to yellow. It's become from neutral into acidic. Now, one more thing, if the bacteria produce gas, then you will be able to see this gas production over here. Okay, so gas, mostly carbon dioxide. Okay, so it will be showing in this Durham tube. If there is no gas like this, okay, so no gas. It's still positive, but there is no gas production. So again, the answer will be glucose. Okay, ferment glucose. If the bacteria can ferment glucose, the broth color will become, I just explained that before. Become what? Become yellow. Okay, this means acidic. So this is positive. This is also positive. Okay, yellow. But this one with gas, the other one without gas. Yellow. Yellow color is an indication that bacteria is fermenting glucose and produce what? Again, I already explained this one. Yellow means acid is produced from that reaction. So positive, positive, acid. Okay. What is the pH indicator used in the phenol red glucose with Durham tube test? Okay. So this is not a tricky question. The key word is already there. So the answer will be the, which one? Yes, the phenol red. What is the role of the Durham tube in the phenol red glucose with a Durham tube test? A, B, C, or D? Okay, remember I mentioned before the Durham tube is used to collect, collect the gas producing from or during fermentation. So this is the tube over here. If there is a gas, then you see the gas formation on the tube. So it's smaller tube inside the bigger tube. Okay, so you see the gas over here. This one is no gas. All bacteria produce gas when they ferment. True or false? We already mentioned that. When they ferment, of course, it's become yellow, but some of them produce gas, some of them is not. So the answer will be false. Okay, so this one produce, okay, so acid plus gas. This one is just acid, no gas production. So this is false. Okay, some ferments without gas, some ferments with gas production. If the bacteria do not ferment the glucose, the phenol red glucose broth will stay what? Yeah, red over here, so it's still red. If it stay red, it means negative. No fermentation, no glucose fermentation. If the bacteria do not ferment glucose, the phenol red glucose broth will change color from red to, okay? So if the bacteria do ferment glucose, so it's positive result, okay? What is the color change? Yes, from red to yellow, okay? This is acidic. Okay, so this is negative, positive, positive. What is indicated by color change of phenol red glucose broth from red to yellow? Again, this is just another question, similar question. So when it is red to yellow, it means that what? Glucose was produced? No. Gas was produced? Not really, because gas is in the Durham tube, not in the color. Okay. Carbon dioxide was produced. Yeah, this is in the Durham tube, so this is should be no. Acid was produced. Yes, so acid is color change, red to yellow. Okay, so this is positive, positive. The color change from red to yellow. A phenol red glucose with Durham tube has been inoculated with bacteria, okay, unknown bacteria, and incubated. After 48 hours or 2 days, the broth color changed to yellow and the Durham tube filled with gas. Interpret the result. Okay, so what is your answer? Remember, yellow means acid. So you have to find the acid first, which is mean that the bacteria fermenting the glucose. The organism fermenting the glucose. Yes, not. So this is not the answer. Fermenting the glucose. Yes. Okay. It's not. So two choice, A or C. But remember, it said that Durham tube filled with gas. So it means that the organism also produce gas. So the correct answer will be A. So this one over here. Okay. Ferment glucose and gas production. A phenol red glucose with Durham tube has been inoculated with bacteria and incubated. After 48 hours, the broth color is changed to yellow, but the Durham tube is not filled with gas. Interpret the result. Of course, it means that fermenting because yellow, but there is no gas, okay, not filled with gas, so it's gonna be this one. So C is the correct answer, which is this over here. C, the correct answer. The methyl red test is a fermentation test used to differentiate many gram-negative bacilli based on the ability to use blank pathway, A or B. So the methyl red test is used to identify bacteria or gram-negative bacteria that are able to use mixed acid pathway. Okay, so they can use lactic acid pathway, acetic, succinate, and also formic acid pathway. And the color will change because of this mixed acid pathway. Which of the following sugar is fermented in the methyl red test? So which sugar fermented for methyl red test? Okay, it's going to be the same, which is glucose. So glucose is the sugar that will be fermented by this methyl red test. So what is the color of positive result of the methyl red test? Okay, changing if it is using mixed acid pathway, the color will change into A, B, C, D. Is your answer? Yes, it is red color. It's become red. So positive result over here is red. Compared to the phenol red test, the positive result is yellow. So red is the positive result. Yellow on doesn't change is negative result. Okay. Yeah, so red or cherry red. What the interpretations of positive result, cherry red, of the methyl red test? A or B? I mentioned before, this is for testing the ability of gram-negative bacillobacteria that able to ferment glucose using mixed acid pathway. Okay, so they use lactic acid pathway. They also use the acetic acid pathway. They use succinic acid pathway and also formic acid pathway. So these are mixed acid pathway, and it will give the red color for positive. What is the color of negative results of the methyl red test? Okay, remember it starts from yellow. So it starts from yellow, it will stay yellow or no color change. So this is negative, stay like this. It is positive, okay, cherry red. What the interpretations of a negative result, yellow or no color change, of the methyl red test, A or B? Of course, if it is positive, they're able to using the mixed acid pathway. If it's negative result, then they only use one pathway, which is 2,3-beta-nediol pathway. And it will not change the pH, it will not change the color. What is the role of methyl red in the methyl red test? What it is used for? Of course, this is an indicator, pH change indicator. If the pH change, then the color will also change. If it is the pH stay the same, the color stay the same, which is yellow in this case. The MacConkey, so this is the next test, it's called the MacConkey agar test, is a blank medium for selecting gram-negative bacilli and differentiating between lactose and non-lactose fermenters. So this MacConkey is used for determining bacteria that are able to ferment lactose. Okay, this one is lactose, not glucose anymore. Okay, MacConkey. So it is medium. What type of medium it is? Selective, differential, general purpose, A or B. Of course, it is not general purpose. This is used for selecting. It means that it's selective, but also the color change is mean that there is a differential, because it can distinguish type of bacteria with another type of bacteria by changing the color. So it is A and B, both selective and differential. If you see over here, this is non-lactose fermenter. This is negative result, and this is the lactose fermenter. This is positive result. The color, the color of the agar will change. Which of the following sugar is fermented in the MacConkey agar test? Yeah, already described that before, which is the lactose. So this is the ability of bacteria, especially gram-negative bacilli, to ferment lactose. Which of the following will form pink colonies on MacConkey agar? A, B, C, or D? Remember, MacConkey is for lactose, so look for the lactose. Okay, it's not glucose, so this one is not correct. Choices. So C or D. Remember, the color is changed. So the color is changed, then the answer will be positive, which is lactose fermenter. Non-lactose fermenter is negative. The color will not change. What is the role of neutral red in the MacConkey agar? What is the role of this neutral red? Yes, this is for pH indicator. If the pH changes, then the agar color will change because of this neutral red reagents. What is the role of crystal violet and bile salt in this MacConkey agar? So there is a crystal red, the crystal violet and bile salts in this agar media, and it is used for what? It is the selective one. Okay, it is for the selective, which is select the in bacteria that's gram positive. So gram positive bacteria will not grow on the media. Therefore, it is only for gram negative bacteria. So gram positive bacteria will die with this crystal violet and the bile salt. So the crystal violet and bile salt will kill gram positive bacteria. It makes the media is become selective because it's select the type of the bacteria that will grow on the media, which is only gram negative bacteria. Okay, it will kill the gram positive bacteria. Which of the following biochemical traits can be determined from observation of colonies on MacConkey agar? Remember, what is the function of MacConkey agar? To know whether bacteria are able to ferment lactose or not. So look for the lactose. So that's the answer. So the ability of bacteria to ferment lactose. The citrate test, this is another test called the citrate test, determines whether a bacterial isolate can use citrate as a blank source, and ammonium cell as a sole blank source. Okay, what is that? A, B, C, or D? So citrate has carbon over it, so it's going to be carbon source. Now, how about the ammonium? Ammonium has nitrogen in it. Okay, so this is nitrogen source. So the answer will be C, carbon and nitrogen. What is the color of the medium if the bacterial isolate can utilize citrate as their carbon source? Choose your answer, A, B, C, or D. Yes, the color will be changed into blue. Okay, so blue means positive. Negative will be green. So as you see over here, this is blue positive, green negative result. See, so from green to blue. What is the color of the medium if the result is negative? Yes, we just mentioned that. Negative is green. Blue is positive. So this is negative, this is positive. From negative to positive. How would a result be reported if the organism grow on citrate agar and the color change to blue? Okay, how you report this result? If the color change to blue, that's the clue, right? So blue means positive. So this means that the citrate positive. So the bacteria are citrate positive bacteria using carbon as a source. Yeah, so this one is positive. How would a result be reported if the organism does not grow on citrate agar and the color stays green. Okay? So it means that these bacteria do not use the carbon and they do not grow on the media and the color stay what? Stay green. It means negative. Okay, so green is negative result. A positive citrate test is indicated by color change of citrate slants from blue to green. Is that correct? No, because this is from green to blue. So it is false. Okay, so it's opposite. Okay, from green to blue. The urea test. So this is the next test. Okay, it's called the urea test. The urea test determines whether the bacterial isolate possess urea and urease enzyme or not. Okay, so this is testing whether the bacteria have this enzyme or not. Is that correct? Yes, it is true. It's testing whether bacteria have urease enzyme or not. So this urease enzyme, okay, will catalyze the reactions of urea into ammonia. Okay, here. So the enzyme is called the urease. Okay, so the productions will be mainly ammonia. Okay, urease enzyme hydrolyze ammonia, forming urea and carbon dioxide. It is true or false? I just mentioned before. Okay, it is urea and then it's produce ammonia plus gas carbon dioxide. So urea with the enzyme, which is urease, will produce ammonia plus carbon dioxide. Okay, so this is opposite. So this is false. Okay. The agar medium for urea test contains what? Okay. What is contents? Of course, this is urea test. It should has urea in it. So urea is the substrate. And carbon dioxide, so this is choice over here. Okay, which one? Carbon dioxide is a result. So this is not contains in the media. This is the result after the process. And to see the process, we need indicator. So indicator will tell you whether the process is positive or not, okay, by changing the color. So we need this, the phenol red as an indicator. So this will be the answer. The function of phenol red in the urea test is as I just mentioned that, A, B, C, or D. This is for pH indicator. So when the pH changes, then the color will change from negative to positive. Now, what is the positive color? That's the question. What is the positive color for this urea test? Yes, the color is bright pink. Okay, so negative is kind of yellow, and when it's bright pink, it's positive. It means that the bacteria has urease, able to hydrolyze the urea into ammonia and carbon dioxide. The presence of urease is detected by what? So we just mentioned that color change over here. So what is happening here? What is the answer? Okay, so look at the bright pink. Oh, all bright pink. So that's all the choice over here. So the color is correct over here. Now, what is happening in here? Whether the pH increase or decrease, that's your choice. It's actually from yellow to pink. It means that the pH increase. So it's become alkaline. Right? So alkaline, then it will pink. Okay, yellow to pink color. So that should be the answer over here. Okay, the pH increase. Hydrolysis of urea result in, we just mentioned that. What is the result? This is the alkaline end products. Okay, pH increase. Okay, above 7, maybe 8, 9, 10. It will change the color. The nitrate test determines an organism to blank nitrate. So what it is? The test is used for A, B, C, or D. Yes, reduce nitrate. So nitrate is NO3. It reduce means the oxygen is taken. So it's become NO2. So this is nitrate. When it is only 2 oxygen, is nitrite. Okay, so this is reduce. Okay, the opposite is oxidize. Okay, from nitrite to nitrate, the oxygen adding is called the oxidize. So the process over here is reduce. It's not the oxidize. Okay. Bacteria that able to reduce nitrate will produce blank as a result. I just mentioned that nitrate reduce into nitrite. Okay, so this should be the answer. Okay, so from 3 oxygen into just 2 oxygens over there. Some bacteria can continue reduce nitrite into NO3 and then NO2 and then what? Okay, so reduce. Okay, so it means that the oxygen is become less. So which one is that? Okay, remember nitrogen gas, there is no oxygen over here. So that is, you see that one of here over here. So that should be the answer. And the other one is going to be ammonia, because ammonia also do not have the oxygens. Okay. What end product is detected by the red color change when nitrate reagents A and B added? A, B, C, or D? Okay, again, this indicate the production of nitrate. So if the color change, okay, red is positive, that the nitrate, okay, the nitrate is A and B added, and then what is the results? It's actually the nitrate. Okay, so the nitrates here. Okay, so it means that the nitrate is present. Okay, the substrate is still there. Oh, that's positive. Oh, positive. Let me see. What end product is detected by the red color change? Red color change when nitrate reagent A and B added. A and B added over here. So by the red color. So the red color changes to kind of blank color. So this is the presence of nitrates. Okay, so sorry. Okay, should be this. Okay, from red to kind of blank or clear color. Okay, so this is the presence of nitrite. Okay, so this should be C. Okay, I'm sorry for this. Okay, so C is the correct answer, nitrate. Now, no color change upon the addition of nitrate reagents A and B indicate that the organism did not reduce nitrate. True or false? This is false. Yeah, because it could be the nitrate is changed into nitrite and then into nitrogen gas or ammonia. So this actually will not give the change of color. Okay? So this is false. Okay, if you see here, the positive results, no change color. Okay? If we put zinc, put zinc over here, okay, and this is, see this E. coli over here, give a positive, is E. coli able to reduce the nitrate into nitrite, okay, but here, over here, the Pseudomonas aeruginosa, they're able to ferment, able to reduce the nitrate, but the result is not nitrite. The result is actually the other one, maybe nitrogens. No color change upon the addition of nitrate reagent A and B could indicate that the organism did not reduce nitrate or nitrate was reduced further into nitrogen gas or ammonia. By adding zinc, it can be detected that if the nitrate was not reduced, the color stay the same. Okay, what is the answer for this one? It should be false. Okay, look at this result over here. Okay, it's actually when the nitrate was not reduced, the color will change into red. Okay, plus precipitation. Okay, it means that this precipitation is actually the nitrate is present. Okay, so it means that the nitrates, which is the substrate, is still there. So it is negative result over here. Okay, so it's false. So changing color is actually negative result. After incubations, the additional nitrate reagent A and B did not produce a color change. So, but you cannot tell whether it is able to reduce nitrate or not. We have to follow the next step, which is addition of zinc. And then if they change color, what happened? So what is the correct interpretation of that? We just mentioned, if you change the color change, it means the nitrate still there, or is not reduced. What does that mean? It means it is negative. Okay, so nitrate was not reduced to nitrate by the microorganisms. Was not reduced by the, was not. So the answer should be this one. So the nitrate is still there, it's not produced by the organism. A bacterial culture is being tested for its ability to reduce nitrate. After incubation and growth of the bacteria is nitrate growth, there is no accumulation of gas in the Durham tube. The test for the presence of nitrate is colorless. But remember, colorless is mean that maybe it is reduced or maybe not. When zinc powder is adding, a bright red color develop. Interpret the result. Okay, so the bright red color develop, then it means that the bacteria reduce nitrate to nitrogen gas or ammonia. Right? So the nitrate is actually produced more. Oh, sorry. Remember the color. So the color means nitrate is still there. Okay, so it means that the bacteria does not produce the nitrate. So the bacteria were unable to reduce nitrate to nitrate. So this is the similar question that, like before. So the color is changed. Bright red color after zinc is mean negative. SIM, sulfide indole motility medium, is a semi-solid medium that can test. So this is another test, okay, continuing test. For what? Hydrogen sulfide production, indole production, or motility of bacteria, or D, all of the above. Okay, the name is telling you it's testing for sulfide, indoles, and motility or movement of bacteria. So the answer will be all of the above. Okay, so this is the result of the test. If the indoles produce, then you see this ring on the top. Okay, that's indole positive. Positive indole. And then there is nothing, so it's negative. And you don't see nothing also, no cloudy is negative, non-motor. Now, if you see this one is negative, so indole is negative. And you see this is the hydrogen sulfide productions. And then the color is not cloudy, so it is non-motil, or this negative for motility. Look at this one. You see the indole, so indole positive. And of course, because the color changes, then it's going to be positive. And you see all cloudy color, it is motil. Okay, so all positive over here. How about this one? So indole is negative. You don't see it. Okay, so indole negative. Of course, this one is going to be positive and also motile. Okay, so that's the interpretations of the result. Okay, so it is all of the above. Okay, now look at the result over here. What caused the blackening of the SIM medium? The blackening is productions of hydrogen sulfide. Okay, so the blackening over here is the productions of hydrogen sulfide. What does the bright pink ring indicate? A, B, C, or D? Okay, remember the ring is related to indole production, which is here and here. Okay, so that's positive result for indole. What does the turbidity or cloudiness indicate? So turbidity or cloudiness indicate the bacteria are motile. or move. Okay? So this one over here, all the medium are cloudy. So it is motility, okay, or movement. How is same medium inoculated? How? A, B, C, or D? So if you see, the result is actually the bacteria is stabbed. Okay? Is stabbed with the inoculating needle. So you put needle that have bacteria in this media, okay, in this agar media. And then this is actually where the bacteria are located. If bacteria stay here, it means that it's not motile. But when everything is cloudy like this, it means that bacteria is moved from this inoculation area, which is the step with the needle, to other area of the medium. Therefore, when you see this cloudiness, it means the bacteria are moving. They are moving. So it's motile. Okay, motile move. How is indole production detected? Indoles, remember this is about ring. So it's a pink ring using the Kovac reagent. So reagent of Kovac will be dropped into the media and then see what happens. If there is a bright pink ring on the top, that means positive indole production. So this one is positive for indole. What is the substrate for indole productions? Now, what is the substrate on that media that will make the ring, okay, the pink ring indole? It is actually amino acid called the tryptophan. So tryptophan is the one that will make this indole after Kovac, okay, after adding Kovac reagents. You have inoculated a tube of SIM, sulfide indole motility medium, and after incubation, the medium remains clear, no cloudy. Okay, so clear. And you can only see grow in the inoculation step. What does this indicate? This indicate that the organism is not moving, is non-motile. Okay, so this one is non-motile. Stay here, stay in the stabbing area. Tryptophan hydrolysis results in A, B, C, or D. Yes, tryptophan plus Kovac reagents. Okay, it will produce the indole. This is ring. Okay, the rings over here is the indole. What must be added to SIM medium to detect tryptophan hydrolysis into indole? Okay, so what we need, the reagent is called the Kovac reagent. So Kovac plus tryptophan will produce this indole. Okay, so this positive result. Bacterial fermentations of sugar usually result in blank end products, whereas the breakdown of proteins and amino acids result in the formation of blank end products. Okay, what is the answer? Okay, sugar, when you ferment the sugar, sugar fermentations, it will produce acidic. It's produce acid. So remember, red to yellow when glucose is fermented. While for the proteins and amino acids, okay, the result is opposite, which is alkaline or base. Okay, so acidic pH, So acidic, the pH is pH lower than 7. Okay. Alkalines pH is higher than 7. Remember, 7 is neutral pH. So the answer will be acidic and then alkaline. Okay, now this testing your ability to interpret the results of your test. Remember, the first thing that you have to do is to isolate pure culture and then perform the gram negative test. I mean, gram stain test, right? Now, after the test, let's say you get gram negative bacilli. So we're gonna use this dichotomous key from gram negative bacilli group. Now, what is the result of the test and how you identify the unknown bacteria? First, it said that the first test is oxidase. Of course, after that is oxidase. It said that oxidase negative. Look for the oxidase negative. So you go this. Okay, you're going here. Okay, and then after this, look at the after negative. What you have to do? Perform this lactose fermentation. Okay, and then look for the lactose fermentation on the result. It said that so lactose is over here. It said that lactose fermentation is positive. Then you have to follow that result. Okay, positive here. Okay, so this is positive. And then what you have to do after that is methyl red. So look at for the methyl red. What it say about the methyl red? Oh, methyl red is negative. So you go here and then here. Right? So methyl red is negative. Go back to the dichotomous T key and it's a negative. Right? What you have to do now is motility test. Look for the motility in the result. Motility. It's over here. Okay, it's going here. You say motility positive and go back to the dichotomous key. It say motility is positive. Now you are able to find the unknown bacteria using that test that you have. Okay, using this results over here. Okay, so this is results given to you. Okay, using this result, then you get the answer is Enterobacter aerogenes, which is C. Okay, so C in the choice. Yes, that's Enterobacter aerogenes. Okay, next one. Okay, we're gonna do the same, perform the same thing. Of course, oxidase is a oxidase negative. Okay, so look at the oxidase negative, which is this. And then what after that? Lactose fermentation. Look for that one. Oh, it's a positive. Then you go positive. And then what is after positive? Methyl red. Look at the methyl red. Oh, methyl red is also positive. Then go to positive. And what is after the methyl red positive? The indole. What is indole? As I am over here. It says indole positive. So indole positive. Then you will find the answer over here, which is E. coli. So D for this unknown bacteria. Okay, next. Okay, another example. Okay, make sure you know how to do it. It's an oxidase negative. So going here. After that is fermentation, lactose fermentation. Look for the lactose. Oh, it's a negative. So go to negative this way. And then under negative, you see the hydrogen sulfide production. Look for the hydrogen sulfide is say positive. So going here, going here, positive this one. And then after positive, look at for the ureas, because that's the next test gonna be. So ureas is positive. Okay, ureas is positive. Then from here, you should be able to know the bacteria is the Proteus vulgaris, which is A. Okay. Yes, so Proteus vulgaris. Hey, next one. Okay, another examples. Make sure you know how to do it. Again, oxidase for negative. So this side, okay, the right side. And then, of course, after that is fermentation of lactose. Look for the lactose. Oh, the lactose fermentation is negative. So you go to this right side, negative. And then under negative is hydrogen sulfide production. Look for that. Oh, this is positive. So you go that one, positive on the left. And then after that, you have to do the urease test. Urease test is negative. Right? Going here is negative on the right side. Then going down, you get the bacteria, which is the Salmonella. Salmonella is F over here. Right? Okay, here you go. Okay, next one. And the next test again. So make sure you're able to use this test. Okay, after gram staining test, then oxidase test, it say that positive. Positive is on the left side over here. And then look for the glucose fermentation now. Glucose, oh, it say negative. So in the right side over here. Under that, you have to perform the nitrate reduction test. Nitrate, oh, it say positive. So it's on the left, on the left side, it's a positive, and it's going down, you find the bacteria, which is Pseudomonas aeruginosa B. Yes. Okay, this is the last one. Okay, again, make sure you are able to use the test, and then by using the test, you will be able to identify the unknown bacteria. Okay, after gram staining, what happens? It is gram negative, and then it said that oxidase positive, which is on the left side. And then after that, what you have to do, look for the glucose fermentation. It's a positive. So on the left side again, glucose positive. And then, oh, you don't have to do any other test because it's already identified the bacteria, which is the Vibrio cholerae. Okay, so that's the answer for that test for identifying the unknown bacteria. I don't know which test number over here, experiment you do with the Vibrio and etc. Remember, there are test number 11 until 20. So there are 10 unknown bacteria that you have to perform on your lab experiment. So again, I suggest you to perform the lab experiments 11 and 20 again and again before taking this exercise. Now, after that, take the exercise for several times until you get the 100 correct answer based on your knowledge. I think that's all. Okay, good luck for your lab exam.